So today we're going to be measuring pH in seawater, one of our fundamental properties for the carbonate system. So the samples we're measuring are these samples here. We have 5 of them from different depths. These were collected on one of the flotsam student cruises on the Walton Smith out in the Florida Straits. So the main idea of what we're doing is we're going to take this water and we're going to add an indicator dye. Chemically, this is one of the most simple labs that we do, where this indicator dye is going to turn different colors depending on the pH of the sample, and we measure that color on the spectrophotometer. We also need temperature and salinity data, so for each of these samples we're going to measure it on the spec. We measure it 3 different wavelengths, and we're going to measure temperature and salinity. So first things first, we need to get each of our samples into our cuvettes. So we're going to be using our long path cuvette cells today so that we can get a really accurate read on that absorbance. So you guys have worked with these before. So we're going to take out both caps, and of course always note-taking. Each of these samples is a unique sample, and so we're going to need to record the number that's on this bottle, and that's going to tell us when we look back at the data logs from the cruise what depth and what day this sample was collected. And then to simplify our life, the cuvettes are already numbered, but we need to keep track of which sample goes into which cuvette. So I've got a list for myself here. This is sample 40. I'm going to pour it into cuvette number 11, so I'll make a note so we keep track. And for this analysis, you want to make sure that you fill the entire cuvette with no bubbles. So we're just going to pour all the way up. I'm working over a bin here so we can overflow a little bit. Now we're going to add our Cresol purple. So bromocresol purple indicator. On here it's written as 0.04% weight per volume. Alternatively, that's roughly a 2 millimolar solution. So we're going to add 20 microliters of that to each sample. We have our pipette set to 20 microliters. Very small volume, so we're going to submerge the tip so the whole thing goes in. And you can see that immediately there's a little bit of purple color where that dye is. Of course, we need to make sure that that gets mixed thoroughly. So we're going to drop our stoppers back in and invert to mix a couple of times. So when we initially added that, you could see this immediate swirl of purple. And now it's all spread out throughout here, so at a glance this doesn't look very colorful, but if we take a look down the long path of the cuvette, which is what the spectrophotometer will be doing, you can see that it's faintly purple. So again, this is why we use tools like a spectrophotometer to measure color, much more effective than our naked eye. So as usual, we want to make sure that all of our optic sides of the cuvette, these 2 ends, are nice and dry before we put that in the spectrophotometer. So that's one sample ready to go. So we're going to do that for all 5 of them. As with pretty much all of our analyses, we want to compare our unknown samples to something that is standardized or known. So in this case, we're going to take a sample of seawater and then we're going to add tris, which is a buffer that actually stabilizes the pH. So we're going to make a mix of seawater and tris, and that's going to be our standardized sample. So we're going to take a sample of Rasmus beach water, sort of our readily accessible water, and then we're going to add 10% of this 10% tris hydrochloric acid mixture. So in the end, we're starting out with 1 molar mixture here, then we're going to add 10% into our standard, so in the end we'll have a 0.1 molar mix. So we're going to make a 50 milliliter mix in a beaker so that then we can pour it into our cuvette. So we're going to get 45 mils of Rasmus beach water over here. All right, so we've got 45 milliliters of Rasmus beach water. We'll put that in our beaker. So this is going to become our buffered standard. And we'll take our pH buffering solution, our tris hydrochloric acid solution. We're going to add 5 milliliters for a 10% dilution. So we've got a large volume pipette set to 5 mils. Take 5 milliliters of that and add that into our seawater. And I'm going to pipette up and down a couple of times and use that to mix these two, since we're working in an open top beaker that we can't cap and invert. So we'll just make sure that's a well mixed mixture. So this is now our standard seawater mixture. So now we're going to treat it just like we did our other samples. So we're going to take a cuvette, it's number 24, and we should ideally rinse our cuvette a little bit before we add in our sample. So we'll pour a little bit, swirl that back and forth, pour it in our waste beaker here. And now we'll fill that all the way up to the top, overflow a tiny bit. And then we need to add our indicator dye, so our bromocresol purple indicator dye, 20 microliters. There we go. And then we'll cap and invert just like we did for our samples. All right, now we have everything racked up and ready to go on the spectrophotometer. We are ready to measure all of our samples now on the spectrophotometer. So we're going to be measuring absorbance for each of our samples that we've got queued up over here. We're going to be measuring them at 3 different wavelengths. So first we need to set the baseline on the instrument, and we do that with a cuvette filled with deionized water. So this is clear, it has no dye added. So this is what we'll use to set the baseline on the instrument. So again, these are each of our 10 centimeter cuvette cells. So we're going to put that in our spectrophotometer, putting it in the optical path of the instrument. And then we need to set it to our first wavelength, which is 434. So we're going to hit go to wavelength. And we're going to enter in 434, hit enter. And then we're going to blank the instrument, and this is what tells the instrument that this is our baseline, that this is a clear sample, and setting that baseline at 0 with the deionized water. And then we'll measure that deionized water just to make sure that now it reads 0, which it does. So now we'll take out our deionized water and we can put in our first sample. So I'm going to start with sample 24, cuvette 24, which was our standard seawater. So we'll put that in, make sure that nests all the way into the holder, and hit read. And then you want to make sure you're taking good notes for all of these values. So here is it's 0.025 for our seawater standard. And we're ready to move on to the next sample. So we'll take out that cuvette. I'll put in cuvette number 11, was our first of our unknown samples, so that was bottle number 40. We can hit read, 0.008 for this one. And then we just move through our whole stack of samples. All right, that's all of our samples at 1 wavelength. So now we're going to repeat that process for our second wavelength. So again, we need to set the instrument, 0 it, or set the baseline with our deionized water. So we'll put that cuvette back in. And now we hit go to wavelength to enter a new wavelength. So we want to measure at 578. And then we're going to blank the instrument again, so this is setting the baseline and telling the instrument that with deionized water, the absorbance at this wavelength is 0. So it's now blanked, and we'll read that sample again just to verify that it's actually reading 0, and it does. So now we're ready to measure our first sample at 578 nanometers. So we'll start again with our standardized seawater, which was in cuvette 24. Make sure we still don't have any fingerprints on the sides of the cuvette. Nest that in there, and we can read that. All right, we have one more wavelength we need to read at, so we're going to reset the spectrophotometer one more time. So we'll take our deionized water again. We'll put that in. We're going to set the instrument now to 730, and this is a control for this measurement. The Cresol purple dye, when it's reacted, shouldn't absorb at 730. So at 730 nanometers, so this is what we use as a control on the measurement, sort of a blank measurement on each individual sample. So we're going to need to blank our instrument one more time at 730. Wait for that to finish. And then, so now this is our deionized water in here. We're going to read that just to verify that we did blank the instrument, and so it's reading 0 with the deionized water. And now we are good to go on the last wavelength for our samples. So our first sample again, we'll take cuvette 24 was our standard seawater. Put that in. Measure, and indeed it has very low absorbance, which is what we were expecting. So it's reading 0.002. So again, we're just going to go through each of our samples. So we have cuvette number 11, and we'll read each of those. And this one is reading negative 0.003. That's fine. Negative values, again, we set the baseline based on deionized water. So this just means that this sample is absorbing less than deionized water. We weren't expecting any of the samples to absorb at this wavelength, so basically this is within error of 0. We record it as negative 0.003, and we will use it in our calculations that way. So we'll just run through each of our samples. Cuvette 50. .006, cuvette 51, .014. One's a little higher. This is why we measure our background, because that's going to help us make our calculations more accurate in the end. Cuvette 16, .004. And our last sample for our last wavelength, cuvette 52. .004. All right, so we have all of the absorbances for each of our cuvettes. Do make sure to take your last cuvette out of the spectrophotometer. And that is the core data that we need to calculate pH for each of these samples. The one last thing that we do need to correct for in all of our calculations is we want to measure the temperature and salinity of each of these samples. So for that, we're going to go back to the original sample bottles that we have here, and we're going to use a conductivity meter to measure that. All right, so we need one last piece of information to complete the lab. We need to measure the temperature and salinity of each of these samples to do the fully executed calculation for pH. So we're going to do that with our conductivity meter here, which also has a temperature probe. And the temperature for these is going to be basically the room temperature that we're in right now. So we're going to take each of our samples. So going back to our original sample bottles here, so our first unknown sample, 40, we're going to just submerge our conductivity probe, make sure that the silver dots are completely submerged in our sample, and then we're going to just hit read and wait for the conductivity reading to stabilize. So the temperature probe stabilizes much more quickly, so it is at 22.5 degrees C. And then we'll just wait for that salinity measurement to stabilize. So that is 38.50 practical salinity units for this sample. So now we're ready to move on to our next sample, and we just need to rinse this probe in between each sample since we're submerging it all the way in that sample. So we use some deionized water and just rinse off that probe. Gently dry it with a Kim wipe. So we'll just continue on down the line to measure each of our samples. For our standard seawater, we had prepared 50 milliliters of this, and we need to be able to submerge that probe. So we're just going to transfer the remaining 20 milliliters that we have here into a narrower vial so it's easier to submerge that probe. So we have a 50 milliliter centrifuge tube vial. We'll just pour that in there, and that just gives us a little bit more height. So now we have our about 20 milliliters left in this centrifuge tube. So this is our standard seawater. We'll just submerge that probe and hit read. So our temperature is still about 22.8 degrees Celsius, and then we're just waiting for the conductivity reading to stabilize to get our measure of salinity. So right now it's reading about 32 practical salinity units. We're just waiting for that to stabilize. All right, so 32.05 practical salinity units. So you can see that's a good bit lower than what we were reading for our unknown samples here, which are about 38 practical salinity units. So that's somewhat unsurprising since our standard here was, we used water that was collected at Rasmus Beach. So that's coastal seawater compared to these unknown samples that were collected in the Florida Straits, which are more representative of open ocean samples. So we can see that difference in salinity between the two.